Well, hello again, everybody. Welcome back as we start digging deep into the animal kingdom, our last kingdom that we need to cover this semester. This chapter is a relatively short one. In fact, it's the sort of shortest presentation of all the presentations that I have for the semester. And so this should not be a very long presentation, certainly not as long as some of the previous presentations, and most definitely not as long as the last two will be as we look at the animal kingdom in total in chapters 33 and 34. But this is an important chapter because it lays the groundwork for where we're going in chapters 33 and 34. You're going to find that there's some redundancy here in this presentation because there are going to be some issues or some ideas presented that you've already seen in the previous presentation for chapter 47. There is some crossover there. But what we're going to do in this chapter is lay out the currently accepted phylogeny of the animal kingdom. And I might, at some key points along the way, touch on some of the changes that have occurred over time, because they're kind of interesting in terms of thinking about how our understanding of the animal kingdom has changed, most notably with the use of DNA technology and gene sequencing, which is probably going to continue to cause us to rethink the animal kingdom, because now with the ability to sequence a far larger range of genes, we should be able to establish, potentially at least, more definitive connections between clades on the basis of shared DNA sequences. Of course, it may turn out to be the exact opposite and may turn out to create a whole lot of confusion about who is connected to who. That's the future. And we will touch on some future directions before we're done today. But let's start out by trying to define a little bit more precisely what we mean by an animal, because while you may think that you know what an animal is, biologically, we need to be a little bit more definitive. So there are some characteristics that we associate with an animal. For a number of them, they are not unique to animals, but as a collection, when we collect all of these features together, they clearly define what an animal actually is and differentiate animals from the other eukaryotic organisms that we've touched on throughout the semester. So let's take a look at the collection of defining characteristics that define what we mean by an animal. So the first thing is to recognize they're heterotrophs. Now, again, this is not a unique feature that's found amongst animals, but it is a feature that unites all animals together. So yes, we have talked about other organisms, most notably fungi and many protists that are also heterotrophs. But as a mode of nutrition, heterotrophism is the way that all animals obtain their energy and their raw materials. So it is a uniting feature as far as animals are concerned. Then when we think in terms of body structure, there are several things that are unique to animals. And we've touched on some of those as we've looked at some of the other kingdoms. So most notably, animals are odd in that their cells lack cell walls. We've seen that fungi have chitin for cell walls, plants have cellulose for cell walls, as do many algae. The idea of having a cell wall is in fact a common feature across a broad diversity of life, which makes animals stand out very distinctively by lacking cell walls. That doesn't mean that they don't have external supporting structures for their cells. It's just not rigid in the way that we think of as a cell wall. So you would have been introduced to the concept of the extracellular matrix in Bio 1. We've touched on the role of the extracellular matrix in embryological development in the previous presentation. So now we need to recognize that the presence of the extracellular matrix and the proteins that make it up, like the fibronectins, and most uniquely, collagen as a connective protein, are also unique to animals. And then we have two tissues that are very much uniquely animals, and that is nerve tissue and muscle tissue, which go a long way to defining what an animal is. And in fact, it's the presence of nerve tissue and muscle tissue that establishes the common origin of animals as a group. A little bit more of that to come shortly. Then in terms of reproduction and development, as we've seen, talking about some of the previous kingdoms, again, animal reproduction is very distinctive when compared to the reproductive processes of the other major eukaryotic kingdoms, the plants and the fungi. Remember then, there again, it is spores that are primarily released into the environment, or are at least the direct product of meiosis, not the gametes. But in animals, the gametes are the direct product of meiosis. So that makes the reproductive process in animals somewhat unique. And then, as we saw in the previous presentation for chapter 47, there are a sequence of 4 stages that we identify with embryological development. Now, the first stage, fertilization and the formation of the zygote, is again not a unique feature of animal reproduction. We've seen that fertilization and the production of the zygote also occurs in the plant kingdom and in the fungi kingdom. So that's not a unique feature as far as animals are concerned, but it's what happens next, the 3 stages that we talked at length about in chapter 47 that are unique to animals. So following fertilization, the formation of the zygote, you get cleavage, which, as you should recall, is the subdivision of the cytoplasm of the egg into smaller and smaller compartments through cleavage. Otherwise, we could refer to it as a modified form of cell division, where you have the division process, but you don't have an intervening growth process, which leads to that subdivision. That type of cell division is unique to animals as part of their developmental process. And of course, we pass through the solid ball of cell stage, the morula, which leads us to the hollow ball of cells, the blastula. Then we have the third distinctive stage, or the second distinctive stage, the third stage of embryological development, gastrulation, where you have this reorganization of the embryo and the emergence of the germ layers, either 2 layers or 3 layers, depending on the type of symmetry that you have associated with the body plan of the adult. So remember again, diploblastic gastrulation, 2 layers of germ tissue is associated with radial symmetry, and triploblastic gastrulation is associated with bilateral symmetry. And we'll talk some more about that as we progress through this presentation. Then we do also, from following on from gastrulation, we have organogenesis. And at this point, with the emergence of the organ systems, many animals, particularly those that we describe as being invertebrate, pass through a larval stage. And in some cases, the type of larval stage actually designates a clade. And we'll see that as we talk about the phylogeny of the animal kingdom a little bit later in this presentation. And so once we progress into organogenesis, this is the last stage of embryological development. You may pass through a larval stage, and eventually that larval stage will undergo further development, out of which will emerge the adult. In some cases, it'll go through some form of metamorphosis, as you'll see when we talk about the invertebrates in chapter 33. There are 2 basic types of metamorphosis. There is incomplete metamorphosis, where the larval stage has a similar appearance to the adult, and as it goes through a sequence of metamorphic steps, it takes on an appearance that means that it looks more and more like the adult. Then you have complete metamorphosis, which you are probably familiar with in terms of a caterpillar turning into a butterfly, where you have a complete destruction of the larval body plan, and then out of that deconstructed larval body plan emerges a new body plan, which is the adult body plan. It involves a complete reorganization of the body plan of the organism from its larval stage to its adult stage. So you do find that metamorphosis being a common feature amongst invertebrates, but as you have also discovered, you do see it in some vertebrates. For instance, a tadpole turning into a frog, or a salamander larval stage turning into the adult larval stage. Those are examples of complete metamorphosis, although in the salamander, you could also argue that it is more like an incomplete metamorphosis, in that the juvenile larval form does have an appearance that looks somewhat like the adult salamander, but there is a complete reorganization of the body, a change in feeding habit, transition from aquatic to terrestrial for most of the salamanders. So in some cases, you could argue that it is an incomplete metamorphosis, but you could also argue that it's complete metamorphosis. It just gets more confusing. Anyway, this is a common feature. And so diagrammed here, we have those stages, the key stages, and this should be familiar to you from looking at chapter 47 in our previous presentation. So this is, again, just a little bit of review from a previous chapter. And again, as I said, there is some crossover as we talk about the nature of the animal kingdom and what defines an animal kingdom. Then one of the things that is most distinctive at the genetic level as far as animals is concerned is the presence of the Hox genes. Now, Hox genes belong to a larger family of genes that we see present in most of the eukaryotes. We do see them in fungi. They've been discovered in yeast, so that's fungi. They've been found in plants, and there is a little mustard plant that has become essentially the lab rat for plant science, called the Arabidopsis thaliana, and master control genes, otherwise known as homeogenes or homeotic genes, have been discovered in plants as well. Hox genes belong to the same large umbrella family, but Hox genes as homeotic genes or master control genes are unique to animals and only found in animals. So the presence of Hox genes has been used recently as a way of defining or creating animals as a monophyletic clade, the fact that the Hox genes are present. Now, having said that, what we do know is that the most basal member of the animal kingdom, the sponges, do not contain Hox genes. But the lack of Hox genes does not mean that sponges are not animals, because the organization of sponges and the presence of certain genetic features and tissue features certainly mean that sponges need to be included amongst the animal kingdom, but they should be placed most basally to the animal kingdom. That has been historically the position of sponges, and with the advent of gene sequencing, that hasn't changed. Although, as you'll see when we talk about the future direction of animal taxonomy and phylogenetics, there is, or at least there has been a couple of studies that have questioned whether sponges truly are the most basal group of organisms. And we'll talk a little bit more about that right at the end of the presentation. So taken together, these are the features that define an animal. They are heterotrophs, they are multicellular, they contain tissues, and particularly uniquely muscle tissues and nerve tissues. They have connective proteins, and particularly collagen as the primary connective protein between cells, is a unique type of protein. They have a unique reproductive process with the stages, with those stages in embryological development we talked about in the previous chapter: cleavage, gastrulation, and organogenesis being key features of animal embryological development that are uniquely animal, and then the role of the Hox genes, defining the body plan of animals and basically being in charge of laying out the body plan. And as we will see along the way, and as I mentioned in the previous presentation with chapter 47, it is the nature of the Hox genes, or more specifically, how many cassettes or copies of the basic set of Hox genes that are possessed by animals that determines what sort of animal body plan you get, and very specifically in the transition from an invertebrate body plan to a vertebrate body plan. Those are major steps along in the evolution of the animal kingdom, and that comes down to Hox genes. So here we will take our first break, and I am actually going to make it a break. I know we haven't been going for too long, but I'm actually going to make it an actual break, and I'm going to cut this section up into some smaller chunks using the concept checks. So here is the concept check question for this break. Summarize the 4 main stages of animal development. What family of control genes unique to animals play an important role in establishing animal body plans? I'll see you on the other side of the break with the answers.